Hello again, Jared Pitts here, and today I'm going to be reading another article by Dr. Ray Peat. This one is called Mitochondria and Mortality. And if you want an article that pretty much sums up the basis of a lot of his ideas and where a lot of the quote-unquote pro-metabolic community is coming from, the bioenergetic community is coming from, this is a fantastic article to listen to. And before I get into the article, I just want to remind you in the description I have links to my website, MissouriHealthCoach.com, and then also I have links to some companies that I purchase products from, and they've given me some codes that I can give out to you all so that you can get a discounted price. Places like Health Natura and Life Giving Store, aspirin, Cascara Sagrada, desiccated thyroid, methylene blue, vitamin K, vitamin D, vitamin A, other things like this. So if you're looking for some companies that take pride in their product, then look no further. The links are in my description. Okay, without further ado, this is how it goes. Diet, exercise, and medicine damaging or repairing respiratory metabolism. Main ideas and contexts. So we have a couple of ideas or words here that we're going to define. So lactic acid and carbon dioxide have opposing effects. The next one, intense exercise damages cells in ways that cumulatively impair metabolism. There is clear evidence that glycolysis, producing lactic acid from glucose, has toxic effects, suppressing respiration and killing cells. Within 5 minutes, exercise lowers the activity of enzymes that oxidize glucose. Diabetes, Alzheimer's disease, and general aging involve increased lactic acid production and accumulated metabolic, mitochondrial damage. The products of glycolysis, lactic acid and pyruvic acid, suppress oxidation of glucose. Next we have adaptation to hypoxia or increased carbon dioxide limits the formation of lactic acid. Muscles are 50% more efficient in the adapted state. Glucose, which forms more carbon dioxide than fat does when oxidized, is metabolized more efficiently than fats, requiring less oxygen. Next, we have lactic acidosis. Lactic acidosis, by suppressing oxidation of glucose, increases oxidation of fats, further suppressing glucose oxidation. And I mention this maybe every article, but a lot of these processes have an amplification type of mechanism so if you aren't actively pursuing better health and trying to get out of these cycles, these physiological responses our bodies have to stress, then they can just perpetuate in these amplification processes. So that's why you're here. Estrogen is harmful to mitochondria. Progesterone is beneficial. Progesterone's brain protective and restorative effects involve mitochondrial actions. Next, we have thyroid hormone, palmitic acid, and light activate a crucial respiratory enzyme, suppressing the formation of lactic acid. Palmitic acid occurs in coconut oil and is formed naturally in animal tissues. Unsaturated oils have the opposite effect. And we know palmitic acid is named as such, coming from palm oil, and it is a long-chain saturated fatty acid. Okay, so those long-chain saturated fatty acids that we're told cause issues, we'll leave it at that, right? Here we're seeing, well, maybe that's not the case, and maybe it's actually unsaturated oils that have that effect. The next one we have is heart failure, shock, and other problems involving excess lactic acid can be treated quote-unquote successfully by poisoning glycolysis with dichloroacetic acid, reducing the production of lactic acid, increasing the oxidation of glucose, and increasing cellular ATP concentration. Thyroid, vitamin B1, biotin, etc. do the same. And we know biotin is B7, and vitamin B1, you might have also heard it called thiamine. Okay, so here we go with some definitions. Glycolysis is the conversion of glucose to lactic acid, providing some usable energy, but many times less than oxidation provides. Okay, so glycolysis being a less effective, less productive form of energy creation. The next word is lactic acid, which is produced by splitting glucose to pyruvic acid, followed by its reduction, is associated with calcium uptake and nitric oxide production, depletes energy, contributing to cell death. Okay, and so when it says by its reduction, what does this mean? You can think of it like this. If you think of the electron transport chain, all right, this energy creation process called oxidative phosphorylation, this is the way that we would preferably create energy. Now, what is happening there? It's in the name, electron transport chain. You're transporting electrons along this quote-unquote chain, right? And what we say is that the last electron acceptor is oxygen. And so what happens to that the one before the last one, right? It's having electrons removed from it by oxygen. Okay? And so it's being oxidized. Okay? So this pyruvic acid is being reduced, meaning that it is actually gaining electrons instead of losing electrons. Okay? So maybe more information than what you want, but this is just, it's important when we talk about energy creation. All right? Just like the electricity in your home, you know, it is electrons moving along this copper wire, typically. You know, same in our body. What is energy? This is electrons from our food. We're getting it and we're moving it along, hopefully, our electron transport chain. And, you know, this is an oversimplification, but in order to produce energy, right? Okay, the next word is Crabtree effect. This is the inhibition of cellular respiration by an excess of glucose. Excess of glucose promotes calcium uptake by cells. Next, we have a Pasteur effect. Inhibition of glycolysis, fermentation by oxygen. Next, we have the Randall effect, the inhibition of the oxidation of glucose by an excess of fatty acids. This lowers metabolic efficiency. Estrogen promotes this effect. Next, we have lactated Ringer's solution. And you can hear it in the name, lactated, right? He's been talking about lactic acid a lot already, so keep that in mind. And this is a salt solution that has / been used to, I don't know if that / is supposed to be in there, okay, that has been used to increase blood volume in treating shock. The lactate was apparently chosen as a buffer in place of bicarbonate, as a matter of convenience rather than physiology. The solution is toxic, partly because it contains the form of lactate produced by bacteria, but our own lactate at higher concentrations produces the same sorts of toxic effect, damaging mitochondria. I have a document that I keep for myself. You know, if I go to the hospital, these are the things that my quote-unquote caretaker needs to keep in mind, right? Which would be hopefully my wife or whoever went to the hospital with me. And one of those things is to avoid lactated Ringers. That's a personal thing, you know, based on what I've read. There are better IV fluids and electrolytes that they could give me, and I would make that request personally. The next word is estrogenic phytotoxins, and these damage mitochondria, kill brain cells, and tofu is associated with dementia. And that word phyto is it means plant, right? From a plant. And so that's why it's referencing tofu here, right? Estrogenic phytotoxins. So tofu, higher in these, killing brain cells, associated with dementia. Okay, let's get into the main bulk of the article. A big setup, so hopefully you're still with me. I'm excited to read through this one. Since reading Warburg's, that's Otto Warburg's, publications in the late 1960s and early 70s, and doing my own research on tissue respiration, I've been convinced that Warburg was on the right track in seeing mitochondrial respiration as the controlling influence in cell differentiation, and in seeing cancer as a reversion to a primitive form of life based on a quote-unquote respiratory defect. Harry Rubin's studies of cells in culture have expanded Warburg's picture of the process of cancerization, showing that genetic changes occur only after the cells have been transformed into cancer. It is now well recognized that defective mitochondrial respiration is a central factor in diseases of muscles, brain, liver, kidneys, and other organs. The common view has been that the mitochondrial defects are produced by genetic defects that are either inherited or acquired and are irreversible. Mitochondria depend on some genes in the nuclear chromosomes, but they also contain some genes, and mutations in these specific mitochondrial genes have been associated with various diseases and with aging. Although these aren't the genes that the cancer establishment has focused on as the quote-unquote cause of cancer, for people interested in the achievements of Warburg and Rubin, it is important to know whether mutations in these mitochondrial genes are the cause of respiratory defects or whether a respiratory defect causes the mutations. Recent research seems to show that physiological problems precede and cause the mutations. Okay, and this is like the classic, cock's crow causing the sun to come up. Did the roosters cock-a-doodle-doo make the sun come up, or maybe it was the other way around, right? And so this is what we're seeing here with cellular mutations. Warburg believed that mitochondria supported specialized cell functions by concentrating themselves in the places where energy is needed. This idea has some interesting implications. For example, when the amount of thyroid hormone is increased, or when the organism adapts to a high altitude, the number of mitochondria increases. But in energy deficient states such as diabetes, they don't. How are these crucial organelles called into existence by the hormone that increases respiration and energy, and also by the hypoxic conditions of high altitudes? In both of these conditions, the availability of oxygen is limiting the ability to produce energy. In both conditions, carbon dioxide concentration in tissue is higher. In one case, because thyroid stimulates its production. In the other, because the Haldane effect limits its loss from the lungs. Okay, and this Haldane effect that he's talking about is just this effect that we see in hemoglobin, in which when it is deoxygenated, right, when you're in a hypoxic condition or high altitude, lower oxygen, it has an increased affinity or ability to carry carbon dioxide. Okay, and if you haven't read much on carbon dioxide or read what Dr. Peat has researched and given to us, we have a lot of articles. I've recorded, I believe, most of his articles on carbon dioxide. I have a CO2 playlist. Feel free to check that out. Could carbon dioxide, a major product of mitochondria, help to call mitochondria into existence? My answer to this is quote-unquote yes. And it will help to briefly explain how I see mitochondria. Although I have no hesitancy in accepting that organelles can be exchanged between species, and that it is conceivable that mitochondria might have been derived from symbiotic bacteria, I am reluctant to believe that something happens just because it could happen. For example, Francis Crick proposed that life on Earth originated when genes arrived here on space dust from some other world. That's a theoretical possibility, but what's the point? It just avoids explaining how the highly organized material came into existence somewhere else. And it probably seriously interfered with the consideration of the ways life could arise here. Similarly, some people like to think that mitochondria and chloroplasts were originally bacteria that came into symbiosis with another kind of living material, consisting of nucleus and cytoplasm. Like Crick's quote-unquote space germs, it can be argued that it's possible, but the problem is that this explanation can stop people from thinking freshly about the nature of the various organelles and how they came to exist. How did cells originate? How did mitochondria originate? Quote-unquote germs. And we we see that all over the place, huh? Just blame the germs. Since I have a view of how cells came to exist under conditions that exist on Earth, I should consider whether that view doesn't also reasonably account for their various components. Sidney Fox's proteinoid microspheres provide a good model for the spontaneous formation of primitive cells. Variations of that idea can account for the formation of organelles such as mitochondria and nuclei within cells, and chromosomes within nuclei. The value of this idea of a self-stimulating process in mitochondrial generation is that it suggests many ways to test the idea experimentally, and it suggests explanations for developmental and pathological processes that otherwise would have no coherent explanation. Proteinoid microspheres and coacervates form by acquiring molecules from solution, condensing them into a separate phase with its own physical properties. At every phase boundary, there are numerous physical forces, especially electronic properties that make each kind of interface different from other kinds. All right, stick with me here. This is getting this is getting heavy and deep. Small changes of pH, temperature of salts and other solutes can alter the interfacial forces, causing particles to dissolve or grow or fragment or to move. In the way that carbon dioxide alters the shape and electrical affinities of hemoglobin and other proteins, I propose that it increases the stability of the mitochondrial coacervate, causing it to quote-unquote recruit additional proteins from its external environment, as well as from its own synthetic machinery, to enlarge both its structure and its functions. In the relative absence of carbon dioxide or excess of alternative solutes and adsorbents, such as lactic acid, the stability of the mitochondrial phase would be decreased, and the mitochondria would be degraded in both structure and function. As the backside of the idea that carbon dioxide stabilizes and activates mitochondria, the idea that lactic acid is involved in the degrading of mitochondria can also be tested experimentally, and it is already supported by a considerable amount of circumstantial evidence. The combination of sensitivity to the environment with a kind of positive feedback or inertia either upward or downward corresponds to what we actually see in mitochondrial physiology and pathology. The Crabtree effect, which is the suppression of respiration by glycolysis, is often described as the simple opposite of the Pasteur effect, in which respiration limits glycolysis to the rate that allows its product to be consumed oxidatively. But the Pasteur effect is a normal sort of control system. When the Pasteur effect fails, as in cancer, there is glycolysis which is relatively independent of respiration, causing sugar to be consumed inefficiently. Embryonic tissues sometimes behave in this manner, leading to the suggestion that glycolysis is closely related to growth. Unlike the logical Pasteur effect, the Crabtree effect tends to lower cellular energy and adaptability. Looking at many situations in which increasing the glucose supply increases lactic acid production and suppresses respiration, leading to maladaptive decrease in cellular energy. Okay, and you can see if you understand cellular respiration why this would be bad, right? Increasing the glucose supply should not increase lactic acid production, right? It should not suppress respiration, okay? And so this Crabtree effect is what it's saying, a maladaptive decrease in cellular energy. This shouldn't be happening. Okay, moving on. I have begun thinking of lactic acid as a toxin. The use of Ringer's lactate solution in medicine has led many people to assume that lactate must be beneficial, or they wouldn't put it in the salt solution that is often used in emergencies. However, I think its use here as a buffer is simply a convenience because of the instability of some bicarbonate solutions. On the organismic level, it is clear that lactic acid is the quote-unquote essence of hyperventilation, and that it produces edema and malfunction on a grand scale. The panic reaction, shock lung, vascular leakiness, brain swelling, and finally, multiple organ failure all can be traced to an excess of lactic acid and the related features of hyperventilated physiology. Otto Warburg apparently thought of lactate as simply a sign of the respiratory defect that characterizes cancer. V.S. Chapote at least hinted at its possible role in turning on the catabolic reactions leading to cancer cachexia, or wasting. I think a good case can be made for lactate as the cause of the respiratory defect in cancer, just as it is usually the immediate cause of the respiratory derangement of hyperventilation on the organismic level. And he might get into this here, but a lot of people, with their misinterpretation of Otto Warburg's research, they think that these cancer cells are fed by sugar. Oh, you're getting too much sugar, you're getting too much sucrose in your diet, and it's feeding these mutated cells, right? But Dr. Peat's saying, no, that's a misinterpretation. Higher lactate levels, by causing this respiratory defect that no longer allow these cells to utilize sugar, utilize glucose in the way that they're supposed to, they're using this glycolysis fermentation process, and they're wasting sugar. Okay, and it's almost like you have a car and you're putting gasoline into it, and it's using this gasoline at a good rate, producing energy so your car can move, and then all of a sudden, let's say for example, you have a hole in your fuel line and a lot of it is leaking out, and so it's still burning all this fuel. Let's say, this is just an example, there's all kinds of holes in this example, but all of a sudden you're wasting fuel and you're using a lot more fuel to create the same amount of energy. So saying that the fuel is the cause of the problem is obviously wrong, right? Saying glucose is the cause of the problem, this is just a bad interpretation of Otto Warburg's research. Now think about, okay, well, if we think gasoline is the problem, we'll stop putting gasoline into the car and see what happens, right? That's what people typically do with the big C word, right? They stop putting glucose into the machine, into the car that is the body. Food for thought. The Crabtree effect is usually thought of as just something that happens in tumors and some tissues that are very active glycolytically, and some bacteria when they are given large amounts of glucose. But when we consider lactate, which is produced by normal tissues when they are deprived of oxygen or are disturbed by the stress reaction, the Crabtree effect becomes a very general thing. The quote-unquote respiratory defect that we can see on the organismic level during hyperventilation is very similar to the quote-unquote systemic Crabtree effect that happens during stress, in which respiration is shut down while glycolysis is activated. Since oxidative metabolism is many times more efficient for producing energy than glycolysis is, it is maladaptive to shut it down during stress. Since the presence of lactate is so commonly considered to be a normal and adaptive response to stress, The shutdown of respiration in the presence of lactate is generally considered to be caused by something else, with lactate being seen as an effect rather than a cause. Nitric oxide and calcium excess have been identified as the main endogenous anti-respiratory factors in stress, though free unsaturated fatty acids are clearly involved too. However, glycolysis and the products of glycolysis, lactate and pyruvate, have been found to have a causal role in the suppression of respiration. It is both a cause and a consequence of the respiratory shutdown, though nitric oxide, calcium, and fatty acids are closely involved. Since lactic acid is produced by the breakdown of glucose, a high level of lactate in the blood means that a large amount of sugar is being consumed. In response, the body mobilizes free fatty acids as an additional source of energy. An increase of free fatty acids suppresses the oxidation of glucose. This is called the Randle effect, or glucose fatty acid cycle, substrate competition cycle, etc. Women with higher estrogen and growth hormone usually have more free fatty acids than men, and during exercise oxidize a higher proportion of fatty acids than men do. This fatty acid exposure, quote-unquote, decreases glucose tolerance and undoubtedly explains women's higher incidence of diabetes. While most fatty acids inhibit the oxidation of glucose without immediately inhibiting glycolysis, palmitic acid is unusual in its inhibition of glycolysis and lactate production without inhibiting oxidation. I assume that this largely has to do with its important function in cardiolipin and cytochrome oxidase. Exercise, like aging, obesity, and diabetes, increases the levels of circulating free fatty acids and lactate, but ordinary activity of an integral sort activates the systems in an organized way, increasing carbon dioxide in circulation and efficiency. Different types of exercise have been identified as destructive or reparative to the mitochondria. Quote-unquote concentric muscular work is said to be restorative to the mitochondria. As I understand it, this means contraction with a load and relaxation without a load. The heart's contraction follows this principle, and this could explain the observation that heart mitochondria don't change in the course of ordinary aging. And so if you think about your regular workout routines, it's contraction with a load and then relaxation with a load, right? It's not really relaxation. You're going back to your starting position, but it is, again, with a load. It doesn't give the muscles a chance to actually relax, right? When a person has an accident or surgery and goes into shock, the degree of lactic acidemia is recognized as an indicator of the severity of the problem. Lactated Ringer solution has been commonly used to treat these people, to restore their blood pressure. But when prompt treatment with lactated Ringer solution has been compared with no early treatment at all, the patients who are not quote-unquote resuscitated do better than those who got the early treatment. And when Ringer's lactate has been compared with various other solutions, synthetic starch solutions, synthetic hemoglobin polymer solution, or simply a concentrated solution of sodium chloride, or salt, right? Those who received the lactate solution did least well. For example, of 8 animals treated with another solution, 8 survived, while among 8 treated with Ringer's lactate, 6 died. Okay, so you can see why I said earlier, this is one of my main things. If I'm going into the hospital, I'm requesting that these not be used on myself. Okay, mitochondrial metabolism is now being seen as the basic problem in aging and several degenerative diseases. The tendency has been to see random genetic deterioration as the driving force behind mitochondrial aging. Genetic repair in mitochondria was assumed not to occur. However, recently 2 kinds of genetic repair have been demonstrated. One in which the DNA strand is repaired, and another in which sound mitochondria are quote-unquote recruited to replace the defective, mutated, quote-unquote old mitochondria. In ordinary nuclear chromosomal genes, DNA repair is well known. The other kind of repair, in which unmutated cells replace the genetically damaged cells, has been commonly observed in the skin of the face. During intense sun exposure, mutant cells accumulate, but after a period in which the skin hasn't been exposed to the damaging radiation, the skin is made up of healthy, quote-unquote, young cells. In the way that the skin can be seen to recover from genetic damage that had been considered to be permanent and cumulative, simply by avoiding the damaging factor. Mitochondrial aging is coming to be seen as both avoidable and repairable. The stressful conditions that physiologically harm mitochondria are now being seen as the probable cause for the mitochondrial genetic defects that accumulate with aging. Stressful exercise, which has been known to cause breakage of the nuclear chromosomes, is now seen to damage mitochondrial genes too. Providing energy while reducing stress seems to be all it takes to reverse the accumulated mitochondrial genetic damage. And I think this term, genetic, is just so culturally ingrained in people that it is so difficult for people to get out of that box understanding how malleable it can be. Fewer mitochondrial problems will be considered to be inherited as we develop an integral view of the ways in which mitochondrial physiology is disrupted. Palmitic acid, which is a major component of the cardiolipin, which regulates the main respiratory enzyme, becomes displaced by polyunsaturated fats as aging progresses. Copper tends to be lost from the same enzyme system, and the state of the water is altered as the energetic processes change. While the flow of carbon dioxide moves from the mitochondrion to the cytoplasm and beyond, tending to remove calcium from the mitochondrion in cell, the flow of lactate and other organic ions into the mitochondrion can produce calcium accumulation in the mitochondrion during conditions in which carbon dioxide synthesis and consequently urea synthesis are depressed and other synthetic processes are changed. Glycolysis produces both pyruvate and lactate, and excessive pyruvate produces almost the same inhibitory effect as lactate. Since the Crabtree effect involves nitric oxide and fatty acids as well as calcium, I think it is reasonable to look for the simplest sort of explanation, instead of trying to experimentally trace all the possible interactions of these substances. A simple physical competition between the products of glycolysis and carbon dioxide for the binding sites, such as lysine, that would amount to a phase change in the mitochondrion. Glucose and apparently glycolysis are required for the production of nitric oxide. As for the accumulation of calcium, at least in some types of cell, and these coordinated changes which lower energy production could be produced by a reduction in carbon dioxide, in a physical change even more basic than the energy level represented by ATP. The use of Krebs cycle substances in the synthesis of amino acids and other products would decrease the formation of CO2, creating a situation in which the system would have two possible states, one of the glycolytic stress state and the other the carbon dioxide producing energy efficient state. Besides the frequently discussed interactions of excessively accumulated iron with the unsaturated fatty acids, producing lipid peroxides and other toxins, the accumulated calcium very probably forms some insoluble soaps with the free fatty acids, which are released even from intracellular fats during stress. The growth of new mitochondria probably occasionally leaves behind such useless materials, combining soaps, iron, and porphyrins remaining from damaged respiratory enzymes. When the background of carbon dioxide is high, circulation and oxygenation tend to prevent the anaerobic glycolysis that produces toxic lactic acid, so that a given level of activity will be harmful or helpful, depending on the level of carbon dioxide being produced at rest. When the background of carbon dioxide is high, circulation and oxygenation tend to prevent the anaerobic glycolysis that produces toxic lactic acid, so that a given level of activity will be harmful or helpful depending on the level of carbon dioxide being produced at rest. Preventively, avoiding foods containing lactic acid such as yogurt and sauerkraut would be helpful, since bacterial lactic acid is much more toxic than the type that we form under stress. Avoiding the stress-promoting antithyroid unsaturated oils is extremely important. And it's interesting how many people listen to Dr. Peat, but this is where they get caught up, right? Yogurt and sauerkraut. Those are like a holy grail when it comes to health and things of this nature. Just the whole alternative health field. These fermented foods have been pushed so hard for so long that very difficult for people to understand this and accept that this is a possibility, right? These bacteria that are producing this lactic acid. Well, if it's bad when our bodies produce it, is it possible also bad when bacteria produce it and then we eat it, right? Okay, their role in diabetes, cancer, and other age-related and degenerative diseases, and I think this includes the estrogen-promoted autoimmune diseases, is well established. Avoiding phytoestrogens and other things that increase estrogen exposure, such as protein deficiency, is important, because estrogen causes increased levels of free fatty acids, increases the tendency to metabolize them at the expense of glucose metabolism, increases the tissue content of unsaturated fatty acids, and inhibits thyroid functions. Light promotes glucose oxidation and is known to activate the key respiratory enzyme. Winter sickness, including lethargy and weight gain, and night stress have to be included within the idea of the quote-unquote respiratory defect, shifting to the anti-respiratory production of lactic acid and damaging the mitochondria. Therapeutically, even powerful toxins that block the glycolytic enzyme can improve functions in a variety of organic disturbances, quote-unquote, associated with, and in parentheses we have caused by, excessive production of lactic acid. Unfortunately, the toxin that has become standard treatment for lactic acidosis, dichloroacetic acid, is a carcinogen and eventually produces liver damage and acidosis. But several non-toxic therapies can do the same things. Palmitate, formed from sugar under the influence of thyroid hormone and found in coconut oil. Vitamin B1 or thiamine, biotin or B7, lipoic acid, carbon dioxide, thyroid, naloxone, acetazolamide, for example. And those were all in bold. Progesterone, by blocking estrogen's disruptive effects on the mitochondria, ranks along with thyroid and a diet free of polyunsaturated fats for importance in mitochondrial maintenance. All right. Man, if you stayed with me through all of that, props to you. There's our references at the bottom. And I hope that helps in understanding what's going on inside of your body, right? Your metabolism. When it's talking about respiration, this is cellular respiration. It's production of ATP energy. I hope this was helpful. Please leave any questions or comments in the comment section. Don't forget to like the video, share with people that need this, aka everybody. And until next time, God bless.